Happy New Year, everyone. Welcome back to our first seminar for 2022. It's my great pleasure to introduce Dr. Ryan Schott. He's a new assistant professor in the Department of Biology here at York. Joined us a year ago, just over a year ago. He does some extremely interesting work, which he's going to talk to us about today. He combines computational, genomic, and experimental approaches to understand how the vertebrate visual system evolves and changes its life history in response to changes in life history and the light environment that the organism lives and sees in. He has done his graduate work at the University of Toronto, an MSc in focusing on vertebrate paleontology, and a PhD focusing on molecular evolution and visionary biology. Following that, he went to the Smithsonian Institute, the National Museum of Natural History, as a postdoctoral researcher, where he studied molecular evolution of visual systems in frogs. And I think he's going to tell us about some of that work here today. So welcome, Ryan, and we're looking forward to your talk. Thank you.
Thank you. Thanks for that introduction. That's great. I'm going to reiterate a couple of those things with a little bit of a brief sketch here about my research career. So I'll tell you a bit about that before I get into the research, but I'm quite excited by this opportunity to tell you all a bit about the work I've been doing as part of my PhD postdoc and now here at York. And this might be a little bit different than we're used to at these CVR Vista seminars, but I hope that you will find it interesting nonetheless to hear a bit about a bit of an evolutionary perspective on vision biology. All right, so I was born and raised in Toronto, actually in East York, not too far from where I live now in Thornhill. And I did my degrees, as Rob mentioned, at the University of Toronto, as well as the Royal Ontario Museum, where I started out my research career as an undergraduate and then a graduate student in David Evans' lab at the ROM. And I was studying dome-headed dinosaurs. And there's a photo of a younger version of me doing fieldwork in southern Alberta. And I studied the systematics and ontogeny of these dome-headed dinosaurs using techniques such as geometric morphometrics and micro CT scanning. And while I enjoyed this research quite a bit, I got more and more interested in getting at the mechanistic basis of diversity. And of course, my paleo system was not ideal for that. And so I finished up the master's and moved on to do a PhD in Belinda Chang's lab at the University of Toronto, where my initial project idea was actually to study the genetic basis of horn development and diversity in these horn lizards, an example which is shown there. But being in a vision lab, I quickly became quite fascinated with the diversity of animal visual systems. And so most of my work has focused on the molecular and cellular evolution of visual systems, and in particular the eye and the photoreceptors within different vertebrate groups. So my initial work focused on reptiles, mainly snakes and geckos, but also other vertebrate lineages, including fishes such as cichlids and mammals like bats. After finishing my PhD, I moved on to do a postdoc at the Smithsonian's National Museum of Natural History, where I joined Raina Bell's lab to continue studying visual system evolution, but with a focus on frogs. And from there, I moved on to my current position as an assistant professor in the Department of Biology here at York. And I can't believe it's already been a year now, having started my position, as Rob mentioned, last January. And it has been quite a weird year to start, working almost completely remotely. But despite the unusual circumstances, I have been working to establish the lab and currently have a great group of undergraduate student researchers and hope to have my first grad student or two starting in the fall. Okay, so now that you have a bit of background on my academic trajectory, I wanted to give you a brief overview of my research interests, which are really driven by natural diversity. And this diversity is largely a result of the different environments that organisms inhabit, which favor different characteristics for reproduction and survival. And so I'm broadly interested in how organisms can adapt to their environments, and in particular, how changes at the molecular level, which could be things like changes to protein structure and function, gene expression, or developmental timing, can produce phenotypic diversity and evolutionary novelty from the cellular through to the organismal level. And to study this, I use a combination of computational approaches such as comparative genomics and transcriptomics, and molecular evolutionary methods, as well as some experimental approaches such as imaging and functional assays. And to study these questions, I use the visual system, which is a really great model system to use because of the direct link it provides between an organism and its environment. And this could take the form of things like finding food, finding mates, or avoiding predators. And if we return to this contrast of different environments, I hope you can appreciate the way that they can vary drastically in their spectral properties. So these differences in the light environments favor different visual adaptations, and this has led to a wide diversity of visual systems. And the example of that is shown with these different pupil shapes across these different animal groups. And so I'm broadly interested in how this diversity has evolved, and more specifically, how different molecular and developmental mechanisms contribute to this diversity. And so my research focuses on answering these questions in two main areas: the mechanisms underlying the evolution of photoreceptors, and in particular, new photoreceptor types in the retina, as well as how the visual system can adapt to different spectral environments and behaviors. And I'll start off with my work exploring the mechanisms underlying photoreceptor evolution. But before we get into that, I'd like to give you some background on the vertebrate visual system, although I'm sure for most of you today, this will be very basic, but this way we're all on the same page. So as you know, vision begins when light enters the eye and is focused on the retina by the lens. And the retina is this multi-layered neural tissue that I've shown here in a view from a light and a scanning electron microscope. And the layers become a bit more apparent if we take a look at this little schematic that I drew. But all we'll need to focus on today are these cells here, the light-sensitive photoreceptors. And so there are two broad classes of retinal photoreceptors in vertebrates. And we have rods and cones. And they are made up of three main sections. We have the outer segment, which contains the visual machinery that converts light into an electrical signal. We have the inner segment, which contains the cellular machinery that helps the cell to run. And then the synaptic terminal, which releases the neurotransmitter that helps to control the transmission of the converted light signal through the retina and eventually to the brain, resulting in visual perception. So the rods and cones of vertebrates have two different roles. The rods perform dim light vision, while the cones perform bright light and, in many cases, color vision. To achieve these different roles, rods and cones differ in their physiologies. So rods are highly light sensitive, but they sacrifice temporal and spatial resolution to achieve this. And they also saturate, meaning that they stop responding under bright light conditions. Cones are more or less the opposite of this, where they are less light sensitive, but this isn't really a problem in bright light environments where there's an abundance of light. And instead, they have high temporal and spatial resolution, and they'll never saturate under natural light conditions. These differences in physiology are driven partly by differences in morphology. What I'd like to take note of here is the large cylindrical outer segment of the rod in comparison to the small tapered outer segment of the cones, as well as the closed discs of the rod and the open discs of the cone. So these discs are actually what contain the proteins that are important for vision, including the visual pigments, which are the molecules that actually absorb and respond to light. So there's an example of a protein structure of a visual pigment there. And the visual pigment is made up of an opsin protein, as well as a light-sensitive chromophore called retinal. So vertebrates typically have one type of rod photoreceptor that has a rod opsin or Rh1 based visual pigment. And this pigment absorbs a characteristic amount of light, which is shown here by this spectral absorbance curve with wavelength on the x-axis here and normalized absorbance on the y. And so visual pigments can generally be identified based on their maximal absorbance or their lambda max, which for rod opsins is usually around 500 nanometers. Vertebrates also have 4 main classes of cone opsin-based pigments, and these each absorb different wavelengths of light. And it is these different absorption curves of cone pigments that actually enable color vision. So using birds, a chicken here as an example, you can see the 4 cone classes of cone pigments and their different wavelengths of absorption that go from the violet here to the red. However, some vertebrate groups, such as snakes and geckos, have reduced visual systems where they have lost visual pigments and so have reduced sensitivity ranges and reduced potential for color vision. And these differences in the function of the rod and cone visual pigments also contributes to the difference in the physiology between these 2 types of photoreceptors. In addition to the visual pigments, the discs also contain a number of other proteins involved in visual signaling, called the phototransduction proteins, an example of which is the transducin, which is shown here. And rods and cones each have some distinct phototransduction proteins, and differences in the function of these proteins also contribute to the differences in the physiology between the rods and the cones. And so this combination of differences in morphology, visual pigments, and phototransduction proteins accounts for a large proportion of the difference in physiological function, as well as adaptation to different light environments. So this duplex retina is what we call this when vertebrates have both rods and cones, and it's highly conserved across vertebrates. However, the proportion of rods and cones can vary greatly, and this is another way that vertebrates can adapt to different light environments, where diurnal species such as squirrels have a high proportion of cones, and nocturnal species like this mouse have many more rods. However, in some groups, this has been taken to the extreme, where only one type of photoreceptor is present. And this is something that we term a simplex retina, such as in some snakes, like this all-rod retina in this nocturnal Texas night snake, or this all-cone retina in a diurnal garter snake. We also see this in geckos, where nocturnal species tend to have all-rod retinas, and diurnal species all-cone retinas, like these shown here. And so I was interested in asking the question, how and why do these simplex retinas evolve? And so we'll start by looking at this in snakes. So in a simplified view, we can split snakes into 3 main groups: the highly fossorial blind snakes and worm snakes, a grade of taxa referred to as Henophidia that contains pythons and boas, and these are primarily nocturnal and fossorial species, and finally Cenophidian snakes at the top here, which are a highly diverse group that occupy a wide variety of ecological niches, including both nocturnal and diurnal species, terrestrial, aquatic, and even fully marine species, as well as fossorial species. These highly fossorial blind snakes and worm snakes have only rudimentary photoreceptors, whereas the pythons and boas have these relatively simple duplex retinas. But it is within Cenophidia that we see a wide diversity of photoreceptor complements, ranging from more complicated duplex retinas in some vipers, all-cone retinas in some diurnal colubrids and elapids, intermediate retinas that are found in some semi-nocturnal and secretive species, including in vipers and colubrids, as well as all-rod retinas that are found in some nocturnal colubrids, like that Texas night snake I showed you earlier. So to explain this variation in different retinal types in snakes as well as in geckos, the comparative ophthalmologist Gordon Walls proposed that rods could evolutionary transition to cones and cones to rods in a process that he called photoreceptor transmutation. And so Walls developed a scenario for photoreceptor transmutation in snakes where he proposed that a duplex retina evolved in some snakes into an all-cone retina. And then this all-cone retina later evolved in some lineages into intermediate retinas in some secretive and semi-nocturnal species. And then in a few highly nocturnal species, all-rod retinas were evolved. However, this process had not been studied from a molecular perspective. So I was initially interested in how this all-cone retina evolved. And so we could form 2 main hypotheses for this. Either the rods were lost and you have this blue cone evolving from the ancestral UV cone, or the ancestral rod cell was transmuted evolutionarily into a cone. If the rods were transmuted into cones, then we could expect to find rod visual pigment, as well as other rod proteins, and we might be able to find hints of cone ancestry in the morphology of some of the photoreceptors in the retina. So using immunohistochemical staining, we showed that rod opsin, Rh1, was actually expressed in photoreceptor outer segments. And the rod-specific transmutation, shown here in green, was present in the inner segment. And these were actually overlapping within single cells, which is shown by that faint yellow staining that you can see here. And we found similar patterns in another species of garter snake, as well as in the more distantly related pine snake. And so we had support for these first 2 points. So we found rod visual pigment, as well as other rod proteins in the photoreceptor cells. But what about the morphology of the cells? So using scanning electron microscopy, we could find no evidence of rods. And as you can see that from the image here, all the cells appear to be cones, even at this higher magnification. So we found no evidence of rods with the SEM. But using transmission electron microscopy at a much higher magnification, we could distinguish between the typical cones that have these open discs, which is shown here, and a second type of cone, cone-like cell anyways, that had a longer, more cylindrical outer segment, a narrower inner segment, and importantly, it had closed discs. So these features suggest that this cell is actually a rod that has been evolutionarily modified to more closely resemble a cone. And so here we had hints of rod morphology. So this provided some pretty strong support for the transmutation hypothesis. And then we further showed that the cone-like rods that have rod opsin actually have some cone-like properties as well. So their physiology may have been shifted towards cone-like function. And so the question remained as to why cone-like rods might evolve. And so this might seem like a disadvantage since it would reduce the ability of dim light vision. However, in snakes, there is this large spectral gap in the cone absorption spectra, which you can kind of see here between the SWS1 and LWS pigments here. So we have this large spectral gap, but a cone-like rod could fill in this gap. And this could improve spectral sensitivity as well as chromatic discrimination, and even provide the basis for trichromatic color vision. And so this might be a useful adaptation in a diurnal species that doesn't really need strong dim light vision. And so that's what we had proposed that has happened in these garter snakes at least. More broadly, this provides an interesting example of the role that historical contingency, in this case the previous loss of some cone pigments that provided that gap in spectral sensitivity, how this historical contingency can have a role in forming new cell types with convergent functionality. All right, so returning to Wall's view of photoreceptor transmutation in snakes, we now have a good idea of how diurnal all-cone retinas have evolved. But what about the broader effects of photoreceptor transmutation across snakes? This is especially interesting if you try to put Wall's linear view of transmutation into a more modern phylogenetic context, because the transitions become very complex quite quickly and pretty hard to work out, partly because of how limited the current data that we have on snake retinas is. But I was still interested in asking what effect photoreceptor transmutation has had on the snake visual system. And so I thought I could use molecular evolutionary analyses of genes involved in vision, specifically the phototransduction genes, to get at this problem. And if you recall that there are some both rod-specific and cone-specific phototransduction genes. And so we might hypothesize that there are functional differences in these proteins that occur with shifts between retinal photoreceptor types, such as between all cone and all rod retinas. So to get at these questions, we assembled a large data set of phototransduction genes across reptiles, including new data extracted from draft genomes, as well as de novo assembled whole eye transcriptomes, as well as data from a novel target capture approach that I designed. And I'll just briefly describe this general approach, as well as some of the specific modifications that I made. So this was a solution-based target capture, which is a method to selectively enrich a portion of the genome for a region of interest through binding with a library of probes. This method had mainly been used for resequencing in model organisms, such as exome sequencing, or for capture of conserved regions for phylogenetics. However, my application presented some unique challenges since I needed to both be able to capture at broad phylogenetic scales, while at the same time capturing complete coding sequences, including sequences that might be highly divergent. And so there are two areas that I focus on to adapt the method for cross-species capture across these divergent species. So this is the probes as well as the assembly method. So in designing the probes, I used multiple reference species and also tiled heavily across the exons in order to maximize the potential for the probes to hybridize to distantly related sequences. I also designed a novel assembly pipeline, which utilized multiple assembly algorithms, as well as multiple reference data sets in order to maximize the recovery and completeness of the coding regions. And when we applied this approach across lizards and snakes, we were able to capture from species that were up to 200 million years divergent from the probes. And so this method can be broadly applied to selectively sequence regions of interest from highly divergent species. All right, so returning back to the phototransduction gene dataset, we targeted 29 phototransduction genes, which were divided into 3 categories: those found in rods shown in blue, cones in red, or both types shown in purple. And I'll use this color scheme for the phototransduction genes throughout the rest of the talk. All right, so we can make a couple of predictions. The first being that transmutation may impose distinct selective pressures on snake phototransduction genes. And so to estimate selective pressure, I use Codon models of molecular evolution implemented in the program PAML. And so these are Markov models, which are stochastic processes in which the probability of change from one state to another depends on the current state only and not on past states. And so the advantages of a codon model include the ability to model biologically important properties of the protein coding sequence, such as the transition to transversion rate ratio, the DNDS ratio, as well as codon usage frequencies. Most importantly for our purposes, though, is the non-synonymous to synonymous rate ratio, also called DNDS or omega, which is a measure of selective constraint. When omega is less than 1, it is indicative of negative selection. And when it equals 1, it indicates neutral selection. And when greater than 1, it's indicating the potential for positive selection. And so this provides an estimate of the selective pressure that is acting on a gene. So there are many different types of codon models, and the ones that I'll focus on today are called Clade models. And these have been very useful for testing for long-term shifts in selection pressures that might be associated with changes in the ecology and function. And so this model estimates 3 classes of sites across the sequence alignment. The first, omega 0, which is shown here, is restricted to be less than 1. So this is the negatively selected site class. The second is omega 1. This is the neutral site class where omega must equal 1. And then the third site class is called the divergent site class, and it is free to vary. Now in the null model, there's just a single divergent class across the tree. But in the alternate model, the divergent site class can be different between 2 or more partitions across the phylogeny. And so the results might look something like this, where we can use the likelihood ratio test to determine if the alternate model is a better fit than the null model, indicating a difference in selective pressure between the 2 partitions of the tree. And significant shifts in selective pressure can indicate functional evolution even when omega is less than 1. And so we could predict that we could predict that there would be elevated omega in snakes relative to other reptiles, and that this difference would be even stronger in Cynophidian snakes in particular, where photoreceptor transmutation has occurred, and that some genes may be even under positive selection. So the way I'm going to show these results is in graphs like this, where the x-axis is omega, and the open and closed circles are the background. In this case, that would be the reptiles, and the foreground, in this case, snakes, as the 2 partitions. And so if there's just one circle, the difference is not significant. But if there are 2 circles with the bar between them, then the estimates of the partitions were significantly different. And the longer the bar, the larger the difference. And with estimates greater than 1, indicating positive selection. And I'll divide the results into 3 groups for rod in blue, cone in red, and genes that are expressed in both photoreceptor types in purple. So the results of these tests were highly significant, with the majority of genes having evidence of a different selective pressure in snakes. And this was most prominent in the cone-specific genes, which you can see in the little bar graph there at the bottom. And these differences become even more pronounced when we compare Synophidian snakes to all other reptiles. We also used a 3-partition model to compare Synophidian snakes to other snakes, in turn to other reptiles. And we see that for most genes, Synophidian snakes have the highest omega value. Right, so this closed circle is the farthest to the right, followed by other snakes in gray and other reptiles in the open white circles. And so this is suggesting that there's quite a strong effect of photoreceptor transmutation on the evolution of these genes. And if we take a look more directly at the effect of photoreceptor transmutation by limiting the comparison to within snakes, we see quite a strong pattern. In particular within these cone-specific genes, which were under significant positive selection. And so there's a question about why is there this focus on cone-specific genes? And what we think this could be due to is repeated reversions to nocturnality that are driving the evolution of rod-like function of cone genes. And so you have cone genes functioning normally in all cone retinas, but with repeated shifts to all rod retinas, where more rod-like functionality of the cone genes is favored, we see these elevated evolutionary rates. Okay, so now we have a bit of a better idea about what is happening in snakes. And so let's shift our focus to talk about geckos. So geckos are a diverse lineage of primarily nocturnal lizards, which is unlike most other lizards, which are diurnal. And these nocturnal geckos tend to have all rod retinas. Or there are several independent transitions to diurnality in geckos, and these lineages have all cone retinas. Walls also had a view of photoreceptor transmutation in geckos, which I'll walk through in schematic form for clarity. And so Walls hypothesized that geckos are derived from ancestors with all cone retinas, which subsequently evolved into all rod retinas in the nocturnal ancestor. And then later independently reverted to all cone retinas in a few different lineages. And so there's good evidence to support that the all rod retina is actually derived from an all cone retina. Taking a closer look at the morphology of nocturnal gecko photoreceptors, we can see that the outer segment discs are largely open, as in cones, and in fact the cells have cone features at all levels of their ultrastructure. They actually only resemble rods in the large size and cylindrical shape of the outer segment. It was also found that nocturnal geckos had only cone visual pigments and other cone-specific phototransduction proteins. But despite this, nocturnal gecko rod-like cones have rod-like physiology and also enable nocturnal color vision at light levels when we and most other animals are actually colorblind. But this was based on fairly limited data, and so I was interested in taking a broader scale look to determine whether geckos actually only have cone-specific phototransduction genes. And also what shifts from an all-rod to all-cone retinas have had on the visual genes. So to answer these questions, I expanded on the previous data set to include gecko genomic data, as well as 2 new whole eye transcriptomes, one from a nocturnal gecko, the tokay gecko, and one from a diurnal anole lizard. And so first, to determine whether geckos use only cone genes, we looked at the expression profile of phototransduction genes in the gecko eye, and we found that 5 rod-specific genes that were previously thought to be lost are all expressed. And so we also found these genes in the gecko genomes, including in the diurnal gecko species. And so these rod genes were still maintained even in diurnal species. You can see these are the genes that were lost there. So there were some that were lost, including RH1, the rod opsin. And so we did confirm that loss in our transcriptome as well as in all of the genomes. And this suggests, though, that these remaining rod genes have actually been co-opted by the cone photoreceptors. This is known to occur in some other vertebrate groups, such as mice, which express rod arrestin and rod kinase in their cones, presumably as a nocturnal adaptation. If we compare the relative expression of rod genes in Gecko to Anolis, we find that the expression levels, while highly variable, are similar enough, and that some rod genes actually have relatively more expression in Gecko than they do in Anolis. Right, so Anolis has only lost a single rod gene here, the CNGB1, and they express rod opsin in the retina, despite actually only having cone-like photoreceptors. When we compared the relative expression levels between Gecko and the anole, one of the first things that stood out to us is the relatively much higher expression of GNAT1, which is shown here, and GNAT1 encodes rod transducin. So we looked for rod transducins in the Gecko retina using immunohistochemistry, and while we were able to detect cone transducin as well as the cone opsin RH2, we were not able to detect any rod transducin. So just shown here is no staining for the rod transducin, despite being able to detect rod transducin in the anole using the same antibody. So it's a little bit unclear what's going on there. The transcript that we found in the eye, despite the higher expression level, was actually only a partial fragment. So it's not clear what role transducin is playing, and it's something we'll have to continue to explore in the future. When comparing relative expression patterns in the Gecko and anole, we can also see a couple other interesting differences. And another that I'll draw your attention to is the much higher relative expression of GRK1, which is the rod opsin kinase in geckos. So this protein initiates the shutoff of light signaling by phosphorylating the activated visual pigment. And shutoff is typically slower in rods, which is partly due to the differences in the activity between GRK1 and GRK7, the cone opsin kinase. So high GRK1 expression could actually increase light sensitivity and contribute to the more rod-like physiology that's observed in Gecko photoreceptor cells. Moving on to look more broadly at the effect of photoreceptor transmutation. To determine what effects photoreceptor transmutation might have had on Gecko phototransduction genes, we compared selective constraint. Excuse me. We compared selective constraint in geckos versus other reptiles. So we know, sorry. Still not fully recovered. So we know from previous studies that snakes did have significant differences for most genes. So we first compared selective constraint between geckos and other reptiles in the absence of snakes. So snakes are excluded from this comparison. And we compared geckos to all other reptiles. This is a similar graph to what I showed previously just for that snake study. And so what we found was that the majority of phototransduction genes in geckos had elevated DNDS or omega compared to other reptiles. And this was again most prominent in the cone-specific genes, which kind of was interesting right off the bat. And again, you can see that from the bar graph there. We also want to include snakes to see if both Synophidian snakes and geckos were under similar selective pressures. And so we compared 3 sets of partitions: geckos versus everything else, snakes, Synophidian snakes versus everything else, and geckos plus Synophidian snakes. So the 2 groups with photoreceptor transmutation together versus everything else in these 3 different partitions here. So after adding snakes, the results comparing geckos to everything else were quite similar. So that was good. And the results for Synophidian snakes were essentially the same as before. But the interesting comparison here was including geckos and Synophidian snakes in the same partition, and those results also looked similar. And this was actually the best fit for the 3 partitions for the majority of genes. So here I've faded out all the comparisons that were not the best fit across rows here. And so most of the genes were actually the best fit in this Gecko and Synophidian partition. And so this indicates that there were parallel shifts in selective pressures in these two groups, despite the independent evolution of all rod and all cone retinas. And this implicates convergent adaptive changes in the underlying molecular machinery of phototransduction. And so more broadly, these results suggest that photoreceptor transmutation can evolve through multiple simultaneous genetic mechanisms, including gene loss and gene co-option, changes in gene expression levels, as well as potential functional protein evolution. And so now we have a good starting point, at least, for how and why simplex retinas have evolved in geckos and snakes. So now I'd like to tell you about some of the work I've done in the Tuitara that I was very lucky to be able to do as part of the genome project for this species. So probably to many of you, the Tuitara looks just like a lizard, or this is actually an ancient lineage that is a sister group to squamates, so that is lizards and snakes. And so this used to be a much more speciose lineage, but the Tuitara is the sole surviving member. In terms of its vision, the Tuitara is very interesting because it is a primarily nocturnal, highly visual predator. And it actually has multiple types of rod-like photoreceptors, a little similar to what is seen in geckos. So you can see there is one cone cell here. And so an early electron microscopy study demonstrated that the rod-like photoreceptors have open discs, suggesting that they are derived from cones through photoreceptor transmutation. However, the molecular aspects of the Tuitara visual system were unknown and actually quite difficult to study due to the endangered and highly protective status of the species. And this is why I was quite fortunate to be able to work on the species as part of the Genome Project, which was conducted in partnership with the Maori people who hold guardianship over the Tuitara populations that were used in the study. So when analyzing the visual genes in the Tuitara genome, the first cool thing that I found was that Tuitara actually have all 5 visual opsin genes, despite having these rod-like photoreceptors. And this suggests that the Tuitara actually have robust color vision, and potentially that they have nocturnal color vision with even greater color discrimination than what's seen in geckos. The next thing that I looked at was what the effect of photoreceptor transmutation had on the evolution of the visual genes. And in particular, I wanted to know if the effect was similar to what we saw in snakes and geckos. And so we looked again at the phototransduction genes. But in this case, we found no evidence for selective shifts across the phototransduction genes in Tuitara. Instead, we found that the visual genes were actually highly conserved across an apparently extremely long time scale. And so I thought I would take a look at this in another way by inferring visual gene loss across amniotes. And so I'm showing here a schematic of an amniote tree, highlighting the lineages thought to be ancestrally nocturnal in blue, and those thought to be ancestrally diurnal in yellow. And if we map inferred visual gene loss across this tree, we can see that Tuitara has one of the lowest rates of visual gene loss across amniotes, despite being primarily nocturnal. Whereas we see a general pattern of higher rates of gene loss in other nocturnal lineages, like mammals and snakes, as well as crocodilians here. And so this implies that the Tuitara visual system is incredibly conserved, and I think this may be a result of the unique life history where juveniles adopt diurnality and arboreality to escape predation from the nocturnal terrestrial adults. So the adults can actually cannibalistically predate on the juveniles. And so this dichotomy in visual ecology may have imposed very strict selective constraint on the Tuitara visual system. Okay, so far we've talked about photoreceptor evolution in a few different groups, snakes, geckos, and Tuitara. But there's one more group that I would like to tell you about today, which are the amphibians, represented here by the frog Xenopus. And so amphibians include frogs, salamanders, and caecilians. And this was the focus of my postdoctoral work, and as well as the major focus of my initial research here at York. And so you can see an expanded view of the amphibian tree of life here. And of particular interest to me is that frogs, as well as some salamanders, possess a novel photoreceptor type that is referred to as a green rod. And this is in addition to the typical vertebrate rod that is called a red rod. So there's the red rod here, and then we have this green rod here, a second type of rod that absorbs at quite a different wavelength of light. And so there's evidence that these green rods, at least in some frogs, enable color vision in dim light conditions, similar to geckos, but perhaps through a completely different mechanism. It is quite unclear how this cell type evolved, but it may have been through photoreceptor transmutation. And so there are two main hypotheses for how these green rods could have evolved. The first is from cones through photoreceptor transmutation, and the second possibility is that they evolved from the existing type of rod that's shown here. So if green rods evolved from the existing rods, we would expect to see rod morphology, rod visual pigment, and other rod proteins. And the typical red rods have closed discs, but if the green rods have open discs, this could indicate a cone ancestry. However, some early electron microscopy work found, in leopard frogs at least, that the outer segments of both rods were similar and had closed discs, which is shown here. But instead, though, the green rod cone inner segments are actually quite similar in terms of the location and morphology. So you can see the green rod inner segment and the cone inner segments are quite similar, and they're found in the same area, unlike the typical red rod, which is found in a different area and has quite a different morphology. So we have a bit of a disconnect here where we have difference between the outer segment and inner segment morphology. Okay, so what about the rod visual pigments and other rod proteins? So this has previously been explored in tiger salamanders. And what the authors of this study found was that, as expected, the typical red rods have a rod opsin and rod transducin. And the blue cones have cone opsin SWS2 and cone transducin. So that, again, as expected. But these green rods actually contain the same cone opsin as the blue cone. So they contain the SWS2 cone opsin, but with a rod transducin. And so again, we have a disconnect here with a cone opsin and a rod transducin. So it still isn't clear how this cell type evolved. And so I'm planning to use single-cell RNA-seq to get at this issue. And we've been identifying some candidate species for this approach, such as the chubby frog shown here, which has a relatively high proportion of green rods, which are normally fairly rare. And this is important because while we could identify rods, telling the green rods from the red rods apart based just on their morphology won't be possible. And so we'll have to isolate all the rods from the retina and then sequence them individually. And then using clustering approaches, we can differentiate between the two populations of rods. And this should allow us to identify the genes that are unique to the red and green rods and help us to get at their evolutionary history. And so we can then combine this with new microscopy studies as well. Again, that will let us determine how these green rods have evolved. We can then compare those results to salamanders and generally across the amphibian tree of life with the goal of determining whether the green rods evolved more than once, since not all frogs and salamanders actually appear to have green rods, at least based on our preliminary data. And so we could utilize a whole genome approach to start to look at this. Longer term, I also plan to return to the Gecko system because I think it's a great system to study the repeated evolution of all cone retinas. And this is a unique opportunity because of the complete absence of rods in geckos. And this is then combined with these multiple independent transitions from rod-like to cone photoreceptors. And this could provide some critical insight into the initial evolution of cones, as well as cone-based visual disease, which can be quite difficult to study using mouse models since mice have these such high rod-dominated retinas. Okay, so now I'd like to switch gears a bit to tell you about some of the work I did as part of my postdoc at the Smithsonian that I have been continuing here at York, looking at visual system adaptation. And so there are several different potential mechanisms of visual adaptation, including opsin loss and duplication. We can also have changes in chromophore usage. So you may recall that the chromophore is the light-sensitive component of visual pigments. Vertebrates can use two different types of chromophores called A1 and A2, and these have different absorbance properties. Changes to the opsin sequence can also affect the absorption spectrum, as well as differential opsin expression, which can affect overall sensitivity, as well as the ability for color discrimination. And finally, we have changes to the cellular content and morphology, which I've gone over in the previous sections. So visual adaptation can occur for multiple reasons, including macroevolutionary transitions, such as in bats that inhabit different environments and that use different methods of echolocation, losing opsins, and marine mammals inhabiting different depths, both losing opsins and also shifting their spectral sensitivities to match the blue shifted light environments that occurs in deeper water. The light environment can also have a strong effect, which is illustrated here in comparison of fish species that inhabit clear versus turbid water. And so these fish species have adapted in these different species using multiple mechanisms, including gene loss, chromophore usage, opsin sequence evolution, and differential opsin expression, all of which to shift their absorption spectra to match these different light environments. And so I'm interested in studying these kind of concepts within frogs. And frogs are of particular interest in terms of their visual systems because of their diverse lifestyles. And this includes species like this nocturnal tree frog with large eyes, different species of burrowing frogs, some of which actually retain very highly developed eyes. We have some fully aquatic frogs with these little tiny eyes, little tiny beady eyes you can see there, as well as at least one frog species that is UV reflective. And so this is a large collaborative project, which the overall goal of which is to integrate spectral analyses with molecular evolution and morphometrics to better understand how frog visual systems have evolved with respect to things like habitat, activity patterns, and life history. Today, though, I'll just focus on two of these aspects, the spectral analyses and molecular evolution. So first, a little background on frog visual systems and photoreceptors. So overall, very little is known about how the visual system varies across frogs, but in those species that have been studied, we typically see these two kinds of rods, the red and green rods that I talked about a bit earlier, as well as a potentially variable number of cones that contain LWS, possibly SWS1, and at least one other opsin that hasn't really been identified. It might actually be the rod opsin expressed in a cone, which would be pretty cool. However, this other visual opsin called RH2 is thought to have been lost early in amphibian evolution. In terms of visual pigments, there are typically three pigments that are detectable, which are shown here and are most likely the LWS rod opsin and the SWS2 cone opsin in the green rods. And there is quite an amount of variation, at least in the small number of species that we had data on before starting this project, that suggests that these spectral absorbances may be evolving across the frog tree of life. And so the first thing we've done to study this was a bit of an opportunistic survey of visual opsins across a sample of frogs. And so this was a collaboration with some colleagues at Stephen F. Austin State University who collected frogs locally in Texas and sequenced visual opsins from retinal mRNA. And then I combined this data with data from available genomic and transcriptomic resources, as well as data on GenBank. And we performed an initial analysis of the diversity and molecular evolution of these visual opsin genes in this sample of frogs. And so we found a few interesting results that I'll highlight. And so first, the frogs consistently express 4 of the 5 visual opsins. And we can kind of confirm the loss of RH2 across a broader sample of frog genomes. So we weren't able to find RH2 in any of our mRNA from the retinal cDNA libraries or from any of the genomes. And so we think that this gene is particularly was lost in frogs. However, we didn't find any support for other gene loss. So SWS1 was found in all species and was under similar selective constraint as the other opsins, which is shown in this graph here, depicting average omega values of the 4 opsins with one of the PAML models, M0. And so this supports the presence of a functional SWS1 photoreceptor at least in our sample of frogs, the presence of which was quite unclear previously. We also found a large amount of variation at known spectral tuning sites. So these are sites in the opsin proteins that when changed are known to affect the spectral absorbance maximum, or lambda max, and so these can be involved in adaptation to different spectral environments. However, predicting the effects of these substitutions in frogs was quite difficult because many of the residues that frogs had turned out to be unique to frogs. So they weren't found in other vertebrates where we kind of knew these sites had an effect, or they had really complex interactions with other sites in the protein. We also found statistically significant evidence for positive selection at a small subset of sites in the RH1 and LWS genes, suggesting the potential for functional adaptation here. Further to this, we found conserved changes in both RH1 and SWS2, which remember is found in the green rods, that suggest potential dim light adaptation. And so together, these results suggest that ecological factors such as habitat, life history, as well as behavior may be driving changes to inurin frog visual systems. And really, this is highlighting the need for additional studies, which is something that we are working on currently. So one final thing that I'd like to highlight from this study that I think may be the coolest result that we found, which is the first evidence for visual opsin gene duplication in an amphibian. And so this was with the duplication of the LWS gene in the African bullfrog Pixecephalus adspersus. And so we found distinct copies of the LWS gene on each sex chromosome. You can see these highlighted in the LWS gene tree here. And because this species has ZW sex determination, this means that the females would have two different copies of LWS and suggests a possibility of sex-specific visual adaptation. And so this is something that was really previously only seen in primates. So this is still something we need to confirm, but I think it's a quite exciting result. And we don't have a great idea what this sex-specific adaptation, if it exists, might be, but we do have some kind of speculation on that. So this might be related to a behavior of the females who swim underwater to avoid smaller males in order to reach and mate with the larger dominant male in a group. And so a second red-shifted LWS pigment could provide a visual advantage in the red-shifted freshwater environments that they inhabit. And this is something that is typically achieved through the use of the A2 chromophore instead of the A1 chromophore in the tadpoles of many species. And we'll get into that shortly, as well as in fully aquatic species such as Xenopus that use that A2 chromophore throughout their whole life cycle. This African bullfrog is also one of a small number of diurnal frog species, which generally require further study in order to evaluate other potential adaptations to bright light and color vision in frogs. Okay, so that was our initial study into frog visual opsins, but at the same time as that, we've been gathering a much larger data set using a combination of whole eye transcriptome sequencing to get visual opsin genes, as well as other visual genes, which we'll then analyze using selection analyses such as the Clade models I introduced earlier, and also microspectrophotometry, or MSP, which is a technique that can be used to measure the absorption spectra of individual photoreceptor cells. And there's an image of my collaborator Ellis, who's the one that does the MSP analyses. And so we are using this data to ask how frog visual systems have adapted to different lifestyles. And I have some preliminary results that I can share from 32 eye transcriptomes and MSP data from about 60 species, but our final data set will be over 100 species. And this is work that an undergraduate intern, Maya Wolfock, assisted with at the National Museum of Natural History. And so we scored and then analyzed data with respect to different traits within frogs, including diurnal versus nocturnal activity patterns, aquatic and semi-aquatic frogs versus those that are not, and then comparison between fossorial and subfossorial species and those that are not, and finally scansorial or arboreal species. And to show an example of what the data looks like, we can see that an arboreal species, shown here by this green tree frog, has a fairly different absorbance spectra compared to the fossorial spadefoot toad shown here. And so this is for the RH1, most likely the RH1 opsin found in the red rods. They also differ in the green rods, which most likely have SWS2, as well as in the red cones, which are most likely LWS. And so you can see that the data from the cones is actually quite messy, and this is mainly due to the small size of frog cones that make measurements quite difficult. Looking more broadly across all sample taxa, you can see a fair amount of variation within each pigment type. And if we zoom in on the SWS divided out into habitat type here, we can see that the scansorial species tend to have a more red-shifted SWS2 pigment. And this is something we'll still need to validate with our full data set and some statistical analysis. We also use Clade models to test for shifts in selective constraint associated with different ecological traits that we examined. And I'll show these results a bit differently than before. And so here the estimate for the null model is shown with this unfilled bar. And the estimate from the alternate model, here comparing diurnal to non-diurnal taxa, is shown with the solid bar. And then the asterisk indicates that that difference was significant. And then if we compare across each of the traits that we analyzed, we find that the best fit overall was for this comparison between aquatic and semi-aquatic species and those that were not. And this is potentially interesting because of the differences between aquatic and terrestrial light environments. And so these sequences in aquatic species may be more constrained due to maintaining this aquatic lifestyle throughout their life cycle. For SWS2, we found significantly elevated omegas in the scansorial and arboreal species. And this again was quite interesting because we saw that red-shifted lambda max in these species with MSP. And so this will be something we need to fully explore with our complete data set. For SWS1, the opsin and photoreceptor type that we know the least about in frogs, we found no significant differences. So this is still a bit of a mystery what exactly this the cell type in this opsin is doing in frogs. For RH1, the Ron opsin, we found fossorial and subfossorial species had more conserved RH1s, so lower omega than other frog species. And this is perhaps due to the higher constraints that might be imposed by their dim light environments. And so overall, this has provided some further preliminary evidence that frog visual systems may be adapted to different ecologies and behaviors. And so I'm really excited to see how things pan out with the full data set. And so as I mentioned, this is a large collaborative project. And so I just wanted to give a quick shout out to the Frog Vision team who's working on this. So this is back from before the pandemic when we were still able to meet in person. And by this point, we probably should have had our next and maybe one after that vision retreat to work on this. And we haven't got to do that in person yet, but hopefully soon. So in addition to looking at the visual opsins, we're also looking at the evolution of other genes that are expressed in the eye. So an example is a project being worked on by a former undergraduate student that I co-supervised at the Smithsonian, Jack Boyette, who's now a grad student at the University of Alabama. And so Jack is studying the evolution of non-visual opsin genes. Non-visual opsins are generally involved in non-image forming tasks, such as calibration of circadian rhythm, regulation of melatonin release, and pupillary responses. And so Jack has found significant evidence for positive selection in a number of these non-visual opsin genes, and he's now exploring how different traits may have influenced our evolution using Clade models, and he's working on writing up a paper for that now. There's also a great group of undergraduate researchers in the lab currently here that are investigating the evolution of phototransduction genes in frogs, although we're still in the early stages, and so I don't have anything specific to share with you today about that. But going forward, I plan to expand the phylogenetic focus of these studies to include the other two groups of amphibians, salamanders and caecilians, to be able to ask more broadly how amphibian visual systems have adapted to different environments and behaviors. And this is because amphibians have independently evolved many similar ecologies, behaviors, and life histories, such as these fully aquatic species shown here, fossorial species, and arboreal species, although there's no arboreal caecilians. And so the goal here being to identify major patterns in visual evolution and development across the amphibian tree and life, using both comparative genomics and transcriptomics, along with these molecular evolutionary analyses, but also want to look at how gene expression and developmental shifts can influence visual system evolution and adaptation as well. And so that kind of leans into the final study that I'd like to quickly tell you about today, which is looking at visual system adaptation to different light environments. So previous studies have looked at differences among species that inhabit different light environments, like that example I showed you earlier between fish species inhabiting the clear water and turbid water. But amphibians provide the opportunity to study this shift in light environments within a single individual's lifetime, because a lot of frogs and some salamanders will shift from an aquatic tadpole to a terrestrial adult frog during their development or ontogeny. And so we're interested in asking, how does visual gene expression vary through time? And so to do this, we set up a differential expression experiment using the southern leopard frog, where we sequenced whole eye transcriptomes from 2 life stages, tadpoles and subadults or juveniles. And these were subjected to either a light treatment, where they were kept in the light for 12 hours before the sample was taken, or a dark treatment, where they were kept in the dark for 12 hours. And so we found good separation of the 2 life stages, as shown in this PCA plot of R-log transform counts. So you can see the tadpole and juvenile frogs there, but the light treatments were not well separated. When we looked transcriptome wide, we found that a large percentage of the genes, so 42%, were actually differentially expressed between the 2 life stages, but only 5% of genes were differentially expressed between the light and dark exposure. Because of this large number of genes, we want to focus in and do an analysis specifically of visual genes expressed in the eye. And so we looked at those comparing juveniles to tadpoles, as well as the light versus dark. And we found a large percentage of the genes, again around 40 to 50%, were differentially expressed between juveniles and tadpoles. But again, the light versus dark treatment had a relatively minor effect. And so I'm just going to highlight a couple of the genes that we thought had the most interesting results. And so one of these was this gene called CYP27C1, which was significantly upregulated in tadpoles. And this gene is interesting because it controls and encodes the protein that controls the shift between the vitamin A1 and the vitamin A2 chromophores. And remember, the shift between these chromophores can result in a red shift, where the A2 chromophore red shifts the visual pigment. This is often found in freshwater species. Right, and so we saw this upregulation in tadpoles, and this corresponds to our MSP measurements from tadpoles having a red-shifted lambda max with an A2 best fit compared to in adults that had a lambda max that was more typical of what we would expect, as well as this A1 best fit. And so we see tadpoles using this different chromophore, presumably to adapt to this red-shifted light environment that they inhabit, and then shifting out of that as they metamorphose into their terrestrial frog stage. We also found differential expression of lens crystallines. So the lens crystallines are the structural proteins of the lens. We found a large number of these genes were actually differentially expressed between tadpoles and juveniles. And so this suggests that there are some differences in lens function between these 2 life stages that are probably related to vision in water versus air. But this is definitely something that we're going to need to explore further, perhaps using some proteomics approaches in the future to really look at how the lens may be changing between these life stages. And finally, we saw some interesting differential expression of visual opsins in both LWS, but I think most interestingly in SWS2, the opsin expressed in the green rods, where we see this upregulation in the juveniles. And so this is interesting because we wouldn't necessarily expect a large amount of green rods to be useful for the tadpole stage, where the tadpoles are generally diurnal and inhabiting that aquatic environment. And so this suggests that there may be an increase in the amount of green rods in the juveniles that could be an adaptation to their terrestrial habitat, or also their shift from their diurnal to nocturnal activity patterns. So more broadly, these results highlight expression differences in a range of visual genes, including the ones that I talked about. And these likely underlie the observed morphological and physiological changes that we actually see in frogs through metamorphosis, and then corresponding to adaptive shifts that improve visual ability in these aquatic versus terrestrial light environments. So in the future, my lab is going to look more broadly across frogs to understand how this visual adaptation occurs with these changes, as well as comparing with different changes in life history. So, for example, comparing species that have both aquatic tadpoles and aquatic adults, like the Xenopus here, as well as species like this poison frog that have an aquatic tadpole and a terrestrial diurnal adult. So here it's shifting aquatic to terrestrial, but not diurnal to nocturnal, as well as species that are aquatic tadpoles and then have arboreal adults. And then I think one of the most things I find most exciting here are some species that actually skip the tadpole stage altogether. So they actually develop directly into tiny froglets. And so they have terrestrial juveniles and terrestrial adults. All right, so just to kind of summarize, the kind of research in my lab is in these two main areas, looking at photoreceptor evolution, as well as visual system adaptation to differences in habitat, activity patterns, life histories, and other ecologies and behaviors. And I'm generally interested in how different molecular and developmental mechanisms contribute to the diversity of visual systems. And so there's a lot of, a number of people that I'd like to thank for this work. So the earlier work was done as part of my PhD in Belinda Chang's lab at the University of Toronto. And there's a large number of co-authors on those studies that I'd like to acknowledge, as well as different funding sources, as well as the Frog Vision team, again, shown here, which is a collaboration across a number of institutions, including the Smithsonian, the Natural History Museum in London, the University of Texas at Arlington, as well as now the California Academy of Sciences, where Raina Bell, who was my postdoc advisor, has currently moved to. And this was the lab at the Smithsonian, Raina's lab, the Bell Lab, who's, as I said, now moved to CalAcad. All right, with that, I'd like to thank you all for listening and for this opportunity to tell you a bit about my research. I think there's some interesting opportunities for collaboration with CVR Vista members, particularly with the all-cone system in geckos, which I'm excited to return to soon to study cone development in more detail and the insights that might provide for better understanding cone diseases, as well as a more generally what a comparative evolutionary perspective and the diversity of animal visual systems can tell us about human and computer vision. So again, thank you, and I would be happy to take any questions.